Okay, now it's up. I can see it now. We can see it now.
Yep, I see it.
All right, thank you. Sorry about that.
You may need to, um, I don't know if that's your computer. It's saying low battery.
Yeah, it is.
You may need to charge that because it looks like it will like shut down.
Yeah, all right. Hang on there.
Okay.
Okay. Let's see. Okay, let's see here. The exam coming up, be open in a few days. Is on the syllabus. Probably open it Sunday and keep it open till Tuesday. And hopefully everybody takes the time, settles in, concentrates and take it. It's the same format. 40 questions, 100 minutes, variety of, well, multiple choice, and probably 5 of the short essays. When answering the essays, I want to give full credit, but some students are just kind of saying phrases and listing. When it says list and describe, put a little more describing in there, and not for me not to interpret that you try to be lenient on giving the points, but please take the time to write sentences and describe. And use the time to go back and really complete your thoughts as much as possible. Okay, Knockdown browser. Please everybody follow the rules, show ID, have a lit room. This is just for the college. Once instilled in everybody, we need to keep up standards of security. And on these kinds of courses, distance learning, it's important to have these the system in place, the lockdown respondus browser. Here's our schedule. We're around midpoint, 7th week. And just to remind us, the exams are 65% of the grade, so something like 13% or something. MindTap 25%, and MindTap, if you're looking at the numbers, it adds up to about 1,000 points. So if you miss 5, 10 points, 15 points along the way, it's not a lot. There's 1,000 points to be got. And those mind tap exercises is for just slowing us down and thinking about the content, and so that you can answer open-ended questions with some confidence and some things to say. Reading a book is reading a textbook is not easy to retain information, so we need other ways to absorb it, to think about it. Taking notes is a good idea. If you do that, you can use the online mind tap with the textbook notes, or I think more effective just physically taking some notes as you're reading electronically, or if you put the video, the audio on, and the book can be read to you at different speeds. There's a bunch of apps in MindTap platform to use. And the the video lectures that are posted, as well as these this a bit more detailed. This is from the Cengage materials that I get along with the book. This is actually from a earlier edition of the book where they had a little more detailed summaries, and in addition, I'm highlighting some things that should be obvious what the important things are. Chapter 6, proteins, amino acids. The digestion and absorption of protein, which we had in chapter 3. This is coming back to it in a little more detail, but similar. You should remember protein mostly digested in the stomach, with that gastrin helping put out HCl, which optimizes the environment for the proteases to work on cutting up that the complex proteins into amino acids or smaller peptides, so that can be absorbed in the major part, that part, the duodenum of this small intestine, where the absorption takes place. And so it had a bunch of that from chapter 3, last exam. Major functions of protein. So this is nicely laid out in the more friendly study guide that that you should look at, and I would give you a hard copy and for you to take notes on if we were in person. But hopefully you can make use of that as best as possible. There's a lot of room on this other study guide, this friendlier one, that you can take notes on as you're doing the last preparation for the test. And I do at the end, and with a asterisk somewhere, I do say that this was just experimenting with AI. This by putting information in. This was used Claude to prepare this this little friendly study guide. It worked out pretty nicely. Other detail for the protein chapter, what's distinguishing about proteins. There's 20 of them that make up, and 20 amino acids that make up all our proteins. Proteins have lots of functions, and you should be able to be ready to answer a question just like that. What are list and describe 3 or 4 major functions of proteins, and you should be able to answer that. The structure, function aspect of proteins is very important. The your primary or sequence of amino acids, secondary, tertiary, and in some cases, quaternary structure of more complex proteins like hemoglobin. Stability of protein. And here, if you want later, you can stop the recording. We are recording this and look at these type of questions. What's why do we consider proteins more complex compared to carbohydrates and lipids? That's an excellent question to be pondering and be ready to answer. Not as much the other ones, but significance of high quality protein. What does that mean? High quality protein, mainly that means to us that it has a good balance of the essential amino acids, those that we can't make from other amino acids. So there's 9 of them that we make those other 11 when we need them. We need pools of amino acids to make all our proteins. We're continually making all kinds of proteins, whether they're enzymes or connective tissue or bone, antibodies, all kinds of things that we make protein for. Energy metabolism. These 2, 7, and 8 are around this kind of topic, energy metabolism. How do we generate the ATP in the end that is the currency that we can run all our functions, all our metabolic reactions? Anabolism, catabolism, breakdown and buildup of whatever that might be. Know about the 2 major energy generating cycles. So that's the one is glycolysis. It's the first. Glycolysis can run anaerobically, and then the one that's we get more out of, more efficient, is the TCA, also known as the Krebs cycle, that we generate more reduction power. That is what's needed to make those ATP and the electron transport system. So have that kind of figure in mind. Take a look at that for a while. That big systems picture that's in the textbook. Yeah, enzymes, coenzymes, cofactors. How do we generate that energy? Is that picture? One of the pictures. What do we get from some kind of burger? Could be a traditional hamburger, could be a turkey burger, could be a Beyond Burger, could be an Impossible Burger. It all has protein, carbohydrate, and fat that our system recognizes and sends that to a system to digest, absorb, and in the end, either make energy, immediate energy, that's carbohydrate, or store it as glycogen, or use fat or store fat for later, or aerobic use. I mean, we're always using a mix of fuels, but mainly carbohydrate. That's the preferred fuel to make our ATP. But there's a blend of fat, and if we run low on carbohydrate, we know that we'd go to more use of fat. The thing with fat, as we can see from this diagram, if we use fat or lipid for getting into the TCA cycle, we notice that we different than py that is pyruvate. Pyruvate could, if we needed to, go in reverse direction. That's a 3-carbon pyruvate to make glucose. If we're low and starved of glucose and need glucose for brain, red blood cells, it's essential for that. We can make glucose. We can't do that with fatty acids. Fatty acids, when they're used for energy, we go to the 2-carbon, the CoA, it goes into TCA cycle, etc., to get ATP. So the difference here is with the fat, we cannot go back to glucose, where if we had pyruvate around, it could go backwards. So the backbone of fat we can use to make glucose if necessary, and certain amino acids can be made into pyruvate that can go back to glucose. Some possible questions that could be asked to answer are these shown here. How do we control energy metabolism? What pathways are shared by the macronutrients? And we just saw that in that last diagram. Oxygen and metabolism. So we mentioned there one of the major pathways for ATP glycolysis is anaerobic. We don't need oxygen for that, but the TCA cycle, the more generating more ATP is aerobic. It's always questions around what of the macronutrients we have on board in the body to make energy, and when one is low, when carbohydrates are low, we tend towards using lipids. If lipids is low, if we're kind of in a starvation or toward a starvation mode, where we'll be using protein. It's not preferred. It's kind of negative if we use protein much for energy, because all protein in the body is doing something. It has a function. So if we use it for energy, we're going to start to lose the function that protein was serving. 8 extends that energy metabolism ideas with thinking about how do we use that energy just to run the body at baseline basal metabolic rate, and then what activity, how much activity are we using energy for, and how much repair are we using energy for in a disease state, how much energy we're going to use for that, and body composition. So this is the BMI is the crude measurement for body composition, and has some correlation with long-term chronic disease risk. Balance. And some questions that could ponder that could show up on the test. Yeah, the BMI is a kind of a gross indicator. It's being supplemented currently with things like waist measurement and some other things. So it's a very crude kind of number because it's very simple. Anybody could measure that with its just height and weight. And chapter 9 is maybe starts to bring together several of concepts into something more practical, a little bit more clinical. And we're thinking about things like the classifications of overweight, obesity, underweight, and with an eye on chronic diseases, especially diabetes and cardiovascular disease and stroke, high blood pressure. Number of fat cells, fat cell size. That was discussed a bit in that lecture by Dr. Amir that was posted. So this question definitely will be on the test. That's something about obesogenic environment. What is it? What are ways to modify environments to have them less of this so-called obesogenic type tendency, and that comes about in places, unfortunately, that's considered food deserts that don't have a variety of healthy food, but more on the processed foods. And another study aid that's very helpful are these study it, real colorful. It used to be in the textbook, it's no longer. So this is they're good. It references parts in the book, but also gives a pulls out the very informative figures from the book, like this one right here that was just shown. It also has some review questions. And I remember from last time, if you looked at it from the first test, I do use a few of these that just appear here because they're good concept questions. Definitely we should know how to answer a question around feasting and fasting, and how does the body adapt to that? Where's the fuel go, or where is it stored? How is it stored? When we start using when we're out of carbohydrate to nourish the brain and red blood cells, to some extent these ketone bodies take the place. They could get through the blood-brain barrier to serve the brain for some amount of time. Still, research is still looking at that, and that's the basis of a few diets, right? That propose this is a way to lose weight and have put our body in a mild state of ketosis, which if it goes too far, could be dangerous because it creates a toward an acidic pH of systems, which is dangerous in the body. Some classifications of body composition. kcals have a question that there'll be one or two questions around being able to do that. It was a bit of a calculation around the number of kcals per macronutrient. And, okay, that was the guest lecture, Dr. Amir. And some of the things that she stressed there, this is some important stuff that what's the risk of chronic diseases from our eating pattern and body composition. These are just a few kind of questions. Could be some of these, but just the type of questions that will be on in the second exam. Sum of all chemical reactions, what's that? We should know all these words by now. Homeostasis was on the last test. Osmosis, that's one of the chemical processes, movement of water across the semi-permeable membrane to equilibrate the concentrations of solutes on both sides of the membrane. So a lot of that takes place across cell membranes, interstitial fluids. Talk more about that in the chapter that focuses a bit more on water. Metabolism, catabolism. So catabolism, the opposite is anabolic processes or building. This is breaking down. So most of our digestion is doing this kind of thing, breaking down, and enzymes help that along. And the process usually is by hydrolysis assisted by enzymes. So that's metabolism. Most of the energy is derived from which of these? So it said this is not preferred, proteins are not preferred. We don't get energy from vitamins and minerals. They serve as cofactors. They help the enzymes along, help in cycles, but we don't get energy directly from them. Glycerol, that's the backbone, right? The 3-carbon backbone to which the three fatty acids are bonded, an ester bond, and that's the triglyceride. So that's not the only place. So really it's here. This is carbohydrate, glucose. All carbohydrates are broken down and to glucose units, even if they're fructose or they're eventually made into glucose to feed into the glycolysis and TCA cycle and fatty acids. So that's where energies come from. And these are the parts of or the units that are broken down from carbohydrate and fat or lipids. So glycolysis, that's the first part of the energy metabolism. This is the anaerobic part, so it does not require oxygen. It doesn't, relative to TCA, does not generate an abundance of energy. It does convert glucose, a 6-carbon carbohydrate, two pyruvates, 3-carbon, 3-carbon molecule. And that's at the end of the glycolysis cycle. Pyruvate then gets turns into, is metabolized, converted to acetyl-CoA, which goes into the TCA cycle. Ammonia as a byproduct that's not from glycolysis. That could be from metabolism of amino acids. From pyruvate to TCA, this is the point where once this happens, then pyruvate, then we can't go backwards to make glucose if necessary. So pyruvate 3-carbon to a 2-carbon acetyl-CoA, and that's metabolically irreversible. So that's why that's important. We that's the normal process when we have abundance of carbohydrate, making pyruvate, then going into the TCA cycle. But if it's limiting and we run out of glucose and pyruvate, then we can't go backwards to make glucose if that's necessary. And going in the opposite direction to make glucose is known as this gluconeogenesis. Complete oxidation, for complete oxidation, acetyl-CoA enters. So complete oxidation, that means production of reducing power, and that's what makes so makes our energy, make our ATP. So we oxidize carbohydrate, we oxidize glucose, and we oxidize, and we put this into the TCA cycle. The Cori cycle, that's brought up. Cori cycle is when we're anaerobic and we're creating lactate. And lactate, if it builds up in muscles too much, it's not good. It's thought to create muscle fatigue. And so it's transported, it's shuttled to the liver, and the liver could regenerate some glucose from the lactate, or put it actually into pyruvate. So lactate into pyruvate in the liver. That's called the Cori cycle. So storage for the future is, if we had a choice among these, it would be the triglycerides. So we digest fats, and mainly it's triglycerides in the diet is the fats, and then we have to break them down to fatty acids and glycerol, and then they're put back together for storing in adipose tissue or the liver, or elsewhere. Elsewhere places we don't really want it to be. Dietary nutrient would most rapidly reverse a state of ketosis in a starving person. So ketosis is the lack of carbohydrate. The preferred route to make energy. Ketone bodies are made from fat, from lipid, and it's to generate molecules, ketones that can get to the brain and red blood cells and feed those with a lack of glucose. Which is considered catabolic. Catabolic is breaking down. So we got to find one that's breaking down. So this, I got to look closely at the question here. In the answers, it says from. So we're looking at the first substance that's stated is from the second one. It's kind of worded tricky. Glucose from glycerol. No, that doesn't happen. Glycerol is three carbon. That would be many steps. Let's see if there's a better one. Pyruvate formation from glucose. Okay. So glucose is the six carbon and pyruvate is the three carbon. And so that actually is what happens. Why do we have that? Amino acid from albumin. Well, that's okay too. No, albumin. Albumin is the larger. So this is wrong. I'm sorry about that. The answer is formation of pyruvate from glucose. That's the breakdown of glucose to form pyruvate. Albumin is a complex, it's a large protein, and it's made from amino acids. Got to correct that. Coenzyme. It's a coenzyme. That helps in metabolism in general. It helps the enzyme. Often they're vitamins. They could be mineral. So it's a small active part of an enzyme that binds to organic reactants. So we know that fats in general average 9 kcals per gram, whereas carbohydrate and proteins, if we would metabolize those, we get 4 kcals per gram. And kcals could be, they're correlated with the amount of ATPs are used or generated, or so they have a correlation with that. So why does fatty acid have that 9 versus 4 kcals per gram? And it's if we think of the structure of fats, fatty acids, they're long chains of carbon that have lots of hydrogens attached to them. 2 less hydrogens if they're a double bond. Some fatty acids have double bonds. Those are the unsaturated, monounsaturated, polyunsaturated fatty acids. It's actually the hydrogen carbon bonds. There's a higher percentage of hydrogen carbon bonds in fats as compared to carbohydrates or proteins. And that's understood as why they're more energy dense. Feature of metabolism of surplus dietary fat. So is all fat mostly stored, promotes increased fatty oxidation, fat spares the breakdown of protein, conversion of excess to fat is inefficient, facilitates the conversion of other nutrients to fat. Well, so it is mostly just stored. It could do this also, spare breakdown of protein, but we're talking about surplus dietary fat. So it is stored. It's not gotten rid of easily. It's not burned more just because more is around. So gram for gram, fat is more. So again, fats are 9 kcals per gram, alcohol is 7, and proteins and carbohydrates are 4 kcals per gram. So kcals is, we may see written on packages or elsewhere with a capital C. That's the same as kcals, 1,000 small calories. So this is one of these rare, but there'll be one or two of a calculation question in the browser. You'll have a calculator to use. It'll be up at the top. It should be very visible. So this is based on what we just said there, the amount of kcals per each of the macronutrients. A diet that's 2,200 kcals, and that's kind of standard. When you know on the food labels, things are based on 2,000 kcals. So this is 2,200 kcals, of which 40% of the energy is fat and 20 is protein. How many grams are carbohydrate? So if you need on the test some scratch paper, you could do that. There's a point where it asks you at the end to show the paper that you used. So because this is a closed book test, you're not permitted to have notes. Should know it enough to just answer. So you can have scratch paper if you want to write this down to figure it out. So this means what? How much is left for carbohydrate? That's 60. So it's 40% is for carbohydrate. So we would multiply this times 40%, this times 40%, and then whatever that is divided by 4. So this 40% of this is 880 kcals. If you divide that by 4, that comes to 220. So you could actually have done that if you concentrate. You could do it in your head, or you can use scratch paper for that. So some sort of question like this, 1 or 2 will be on the test. Gluconeogenesis, the production of glucose from non-carbohydrate source. That's the liver. Liver is an amazing organ. There's so many roles. So this is a body composition question. Visceral fat is chiefly stored around abdominal organs, seen as the unhealthy way to store fat. Just being close to organs, it could be spillage around or leakage into or just compromising the functions of organs, in particular liver. Okay. Which of these is possible? It's saying in a cell. Pyruvate to urea, that wouldn't happen. Urea has nitrogen in it, and pyruvate is a straight hydrocarbon. Lactate to pyruvate. Whoop. Okay, I'll put back here. All right. So it is lactate to pyruvate. That's part of the Cori cycle. What the Cori cycle does. Uh, the part of the cell, the organelle of the cell, the powerhouse of the cell is mitochondria. Um, so what's the opposite of mitochondria? It's absolutochondria. Okay, I hope someone's laughing.
I got that
Which
one.
thank you, thank you. Which statement is true about body mass index? Okay, so that's just using the BMI, using height and mass or weight, correlates uh, with disease risk, increases by 1 unit every 10 years, provides estimate of fat, person's height divided by the square of the weight, and its the absolute value is less important than the rate of change. So it is does correlate with risk. Now, it's so the numbers we know go from the teens up to 40 plus. 40 plus is in what we're considering obesity and levels of obesity, and kind of what's called normal is in the 22 to 24 range of this calculation. So it's so this is right in height is in what meters, and well, the numbers are in the between the teens and 40. Measure the rate of nutrient absorption and use by the body. Okay, the rate at which is, if we look at these, and the one we're familiar with is bioavailability. That's part of protein quality, is how bioavailable is the protein to be metabolized. That is broken into absorbable units, which are the amino acids and small peptides. Okay. So that's through those questions. I hope it was been informative to go through them, and just pointing out that study guides, there's really 3 different study guides that could be helpful. The study it or study ITs, which are in each of the modules. Oh no, that's found in the additional materials resources, additional resources. And those more detailed chapter study guides, and this one that I had Claude help me put together. It's kind of nice. It's less detailed, but a nice kind of final, friendly study guide to go through. And if there's any questions or comments, please let me know if you have them now, if you want to speak up, or certainly communicate me by email. And I just want to remind everybody that, and I posted this on Mondays. I get to the tech building about 5:30, sometimes a little earlier. And I'm there if you do want to talk to me in person. It's in room 219 of tech. It's best if you, yes, yeah, that the test is, yes, 40 questions in all, just like the first one. Majority multiple choice, 35, and probably 5 essays, a little less than last test. Last test, I think there was 7. And there was some extra credit. So there'll be some extra credit ones that you want to take a shot at. If that you can't lose, you don't lose points. It's just some additional material just to allow you to increase the grade a bit.
How many extra credit questions are there going to be?
Oh, okay. So let's see, 5. So I think either 2 or 3. 2 or
Thank you.
3. So that's a possibility of 10 or 15 points to make up for if you didn't know some other things. And please, again, I'll just remind you that be as complete and written in full sentences, and don't assume I can fill in your whole thought. Some people really do a really nice job of even highlighting little some part of it and then writing a little paragraph and formatting very nicely. That's not necessary. That's nice. It makes it easier for me to read and give points. But paragraphs and a couple sentence or even a sentence and a space, paragraphs are nice so that I can easily read. And you can just see how when it's clear, I'm looking for certain phrases and ideas. I do want to mention that when all grades, and I think all grades are done, when they're done, then you are able to click into your last exam and this upcoming one to see what the ideal answer, what I had typed or used as what I'm using as a guidance for giving the points. So you'll see your answer, you'll see the correct answer. But for the short answer questions, there's more than what was really needed to get the 5 points for the short answer question. But just want to remind you there that you can click into your grade and get to see how you got the score that you earned, what you got. And if you want to discuss any part of that test, just let me know. We could discuss just by email, or could arrange a WebEx like this, or you can, if you're on campus, you can schedule to see me. I teach on a chemistry course in tech on Monday and Wednesday evenings, 6:30. Any other comments, questions? Okay. All right, sorry for the delay there, fussing around, but something changed with updating WebEx or something. I had to download, and it asked for some other permissions and things, so sorry about that delay. Okay, thanks for joining. I'll post this, it was recorded, my part of it, that it could be seen again or looked at, parts of it or whatever. Okay, good luck, on the next test and studying. It'll be open Sunday probably night, and open, Monday, Tuesday, and I'll see how people are doing. And maybe even into Wednesday. Sometimes I have to remind some people that might have forgotten about the test. Yes, Zorn or B, have a question?
No, you just answered it. I was gonna ask because I noticed that you extended it last time, or you want to extend it again, but you just said you might.
Yeah,